science enthusiasts. Welcome to Science Chat on Twitter. We are triple casting today. We're on Wisdom and Clubhouse. I see some people listening on all the platforms, so hello. And our main platform, of course, is Twitter. Hello, everybody. It's so good to see you all. Every week, we bring an amazing guest to chat with you about their area of knowledge. Today, we have Dr. Sam Horrell, who's a beamline scientist, and we're going to get into all kinds of funky molecular movie stuff. I'm excited, and I don't understand any of it, so I'm glad you're here with us today, Doc. How are you doing?
Yeah, very well, thanks. Thanks for having me.
Yes, I'm excited that you said yes to such short notice, and you were willing to be up very, very early in the morning because you're not in North America. Where are you in the world?
I'm currently in a small town just outside of Oxford called Abingdon. So it's currently 2 in the morning.
Oh, my goodness. You're such a trooper. You are such a trooper. Everybody in the chat, if you have the ability to throw some emojis, give the doc some clapping or something like that. You know, staying up that
late. Well, I went to sleep and set an alarm.
So I introduced you as Dr. Sam Horrell. What's your doctorate in? What's your education in, Doc?
So my doctorate is in biological physics. So I studied at the University of Liverpool, but I was also joint with the place I actually work currently. So it's Diamond Light Source, which is a synchrotron particle accelerator just outside of Oxford.
Oh my goodness. A particle accelerator. Yeah, we'll get to that probably. Pursuing a PhD is nothing for the faint of heart. What got you into science? What was the bug that kept you going?
I've been thinking about this actually since you sent the list of questions. And yeah, I'm not sure there was one specific thing that really got me into science. I remember really enjoying science in school. And one of the very first experiments I remember doing in school was like in year 3 or 4, I think. And it was, we took lots of different liquids and we stuck them in the freezer and saw what froze and what didn't overnight.
Okay.
And I remember washing up liquid didn't freeze, and that was just very fascinating to me.
Was it the question why? Were you curious? Was that it? Like you're just like, why?
Yeah, pretty much. So it was just like, yeah, one of the early things I really remember about school was like, why did this thing freeze and this thing didn't, and all of that sort of stuff.
Gotcha. Okay, so
yeah, like,
go ahead.
Yeah, neither of my parents are scientists. I was the first person of my family to go to university, so yeah, really took the academia a bit too far, I think, in terms of my family going to university and really sticking with it.
So you did start in biochemistry. Was that your undergrad, Doc?
Yes.
Oh,
yeah,
you know what? That's what my undergrad is biochemistry as well.
Very cool.
Yeah, I didn't go past undergrad. I have 2 undergrads, but my specialty is in biochem.
Okay, yeah. So initially, actually, I first applied for medicine.
Oh, okay.
Because I knew I wanted to do something science related, and I didn't really have a good idea of what a science career was. So I applied for medicine, didn't get into any medicine courses. But the University of Leicester offered me a medical biochemistry course.
Okay.
Yeah, so I ended up taking that instead.
Okay, so I have a question.
Yeah.
How do you go from that to a particle accelerator? What happened there? Could you tell us?
Yes, I mean, if you told me at the end of my 3rd year of university that I'd be working in multiple different particle accelerators by the time I was 30, I don't think I would have believed you. So yeah, I don't know how it is for you guys in North America, but for our 3rd year of university, we do a lab project typically.
Mm-hmm.
And my lab project was with a structural biology group at the University of Leicester. So I started purifying proteins for them for both NMR and protein crystallography.
Okay.
And that's kind of how I ended up in starting out in the field.
Okay, so that's the linkage. Hey, you're something you did in your third year. Now, what you do now is, I did some background on it, and I'm still a little fuzzy.
Sure.
Okay, protein crystallography, beamline, and you make movies of proteins in action.
Yeah.
Would you be able to break that down for us?
Sure. So protein crystallography, I guess it's all about finding out how the protein looks like on a atomic level.
Oh, because they're so weird shaped, right? They're super bizarrely shaped.
Yeah, I mean, there's all sorts of shapes and sizes of proteins. Like the very first proteins solved by X-ray crystallography was in the 1960s. So they were much more simple. Like insulin was one of the first, hemoglobin, myoglobin. Nowadays, we're solving structures of like the ribosome, like huge molecular machines. So the field has kind of really jumped from there. Like, but yeah, essentially the way we normally tell people to think about it is like the synchrotron, the particle accelerator, is kind of like a giant microscope. So what you're trying to do is shine light on these proteins, and then you're kind of focusing the light, the X-rays, to then get the resulting structure. But unlike a typical microscope where you have a lens which you can focus to get a clear image, we're using complicated maths and a diffraction pattern instead of our sort of image that we can see with our eyes.
Right, because when I think of a particle accelerator, I think of, particles hitting each other and exploding into cool bits. This is a little different, correct?
Yeah, so our particle accelerator is more of an accelerator and less of a collider. So a lot of people, when I say I work at a particle accelerator, their first thing is, oh, is it like CERN? So CERN is obviously an amazing feat of science. Like it's, 14 kilometers diameter or circumference? I can never remember. But Diamond, where I work, is much smaller. It has a 561 meter circumference ring. And what it does is it produces a beam of electrons, which it forces around a ring using different magnets. And when you take that electron beam and force it to change direction, the electrons give off X-rays in the direction the electrons were traveling. And we then focus those X-rays down to a small beam, and then fire them at our protein crystals.
And does that leave? How do you figure out the shape from that? Is it like an imprint? Like you don't see the electrons because they get stuck in the protein?
So at that point, the electrons have changed direction and are continuing whizzing round the ring.
Okay. Oh, right. I'm sorry. Okay. Yeah.
The X-rays carry on down the beamlines, so the place where I work. And then we have various mirrors and lenses that focus those X-rays and like select out a single wavelength in our case. And then when the X-rays hit the protein crystal, they produce this phenomenon called diffraction. So diffraction is like this beautiful spot pattern. If you Google protein diffraction, you'll see these beautiful sweeping, they're called lunes of spots. And those spots are effectively a representation of, it's called reciprocal space. So if you imagine the protein itself is real space, and the X-rays hit the crystal, scatter, give us a representation of reciprocal space. We then use the maths to focus that reciprocal space back into real space. And that gives us our protein structure.
Now, the question some people are going to be thinking about that I should be asking you is, why is that important?
Mm-hmm.
Why? That's always the why, right? So that's my question to you. Why is that important to know the shape of proteins?
This is Discovery Matters, a collection of stories and insights on matters of discovery that advance life sciences.
Brought to you by the people at Cytiva and driven by our curiosity for all things science. And we've actually heard that it's got real health benefits.
One listener told us that she started running faster thanks to listening to us.
So do it for your health or for your curiosity.
Either way, give us a listen and a rating.
Yeah, I mean, it's a very valid question. Like, Diamond, where I work, is completely government funded, so we are civil servants. Technically, we're all taxpayer funded. So, wanting to know where all of that money is going is a very valid question. But yeah, it's essentially, once you know how the protein looks, you can like the typical phrase is structure leads to function. So once we have a good idea of the structure of our protein, we can figure out how it works. And then from that, say it's a protein involved in a particular factor of a bacteria that infects people, or a virus, or maybe a cancer protein. If we know how it works, we can either design a drug to stop it working or make it work more, depending what the problem might be. So experimental drug design is sort of a big factor of protein crystallography. But then there's also just kind of the basic research side of it. So we want to be able to just see how these proteins work, and then that will kind of feed into other more applicable regions of science, where it actually leads to like a product or a treatment or something like that.
If you're joining us, this is Science Chat. We're triple casting to Wisdom, Clubhouse, and here in Twitter Spaces. On Twitter, I can post stuff into the nest, and there's a picture of protein diffraction. Is that a good photo for people to look at? I don't know if that showed up on your phone yet there, Doc. Protein diffractions, where that's perfect. Gives people the idea of the X-rays and then the shapes that the computer makes from that. The pattern.
Yeah, so when those X-rays hit that protein crystal and you get that spot pattern, so that will be a partial part of the total diffraction that can be produced. So what we do is we'll take that crystal and we'll actually rotate it within the X-ray beam. And depending on how the protein is packed within that crystal, we'll get a complete data set from which we can solve our protein structure.
Right, because they're so twisty and weird, right? Like they're not uniform. Mm-hmm. I see, right? Okay. It's like my hair during COVID. Like every angle of my head, I looked like a different kind of hobbit. It was just outrageous. I know your profile picture, you've got long hair too. So mine wasn't quite that long, yes. But during COVID, my hair just grew up. Like it just grew straight up. Yeah, it was a whole thing. Anyways, on your profile, I was going to ask you this. I know I didn't send this to you ahead of time. It said you're a coronavirus structural task force member.
Yes.
So, like, is that some secret organization like the Illuminati? We have to worry about the Scarlet Witch coming to take us out. It sounds like a super cool team.
Yeah, so it's it started not long after the pandemic, really. So when after the sequence, the genome of the coronavirus had been released, and a couple of groups in China started producing these X-ray crystal structures of coronavirus proteins, a lot of people in the structural biology world jumped on and also started producing structures to a point where it was a lot of structures coming out very quick. And the task force was kind of put together to try and check all of those structures as they were coming out, to make sure that the structures were as good as they could possibly be. Because a lot of the time with these structures, they're published on something called the Protein Data Bank, and they can be used by anyone. They're all free to access, but not everyone that uses those structures is a structural biologist. You get a lot of secondary users who don't necessarily know what constitutes a good protein structure or a bad protein structure. So we wanted to make sure that the computational chemists and stuff that were using these structures were getting the best possible data to start with for doing their
so you comb through it for bad
stuff like that.
You kind of comb through it to get out the bad. Okay. All right.
Yeah, and we also started producing blog posts about all the different proteins, other things about coronavirus. I wrote a blog post on if coronavirus was made in a lab or not. It was all brought together by a brilliant scientist called Andrea Thorn. She's she's working in Hamburg, where I used to work currently. And she kind of brought together, mostly actually the team is made up of undergrad students. So it's a couple of senior academics like me, Andrea, and Gianluca, and then all the rest most of the rest of that is like PhD students, the odd postdoc, and then lots and lots of undergrad students who jumped on to help out.
Yeah, because if any of these structures are wonky, that goes on to further mess up whatever experiment they're going to base the structure on, possibly treatment in the future, right? That's the whole idea.
Yeah, exactly.
You start with, okay.
Yeah. It's like it's bad data in, bad data out, really. Yeah. Like if someone takes one of these PDB structures, protein data bank files, and they there's lots of small details that you might miss. Like there's because our when we collect our data from our crystal, that spot pattern is actually like an average of millions of protein molecules within that crystal. So you can get kind of it's a thing called occupancy within the crystal. So you can have multiple states within the same structure. And sometimes if you're looking to do a simulation, some of the amino acids might have a lower occupancy and might not actually be there all the time. So it's a bit of a minefield just going and downloading a protein data bank file if you don't necessarily know exactly what you're doing with it.
That's like me every time I download some 3D printing file off the internet. I'm like, I hope this works. It says it's Magneto's helmet. Uh-oh. No, it's not. It's a Deadpool face.
Well, on that vein, we do actually have a 3D printable file of the whole coronavirus. So you can print your own structurally accurate coronavirus at home.
Oh, cool.
And put it together. And if you go on, so the website is insidecorona.net.
Okay.
And you can find access to our GitHub repository, which has all the structures that we've looked through and validated. And then there's also a section on there about printing your own virus.
Okay, I'm bringing it up on my computer as we're talking. So I've got 3 more questions before we open up the floor to our wonderful community to ask questions of you, Doc. And I'm scared to ask you this, but what did you think of what did you write in your blog post about was coronavirus made in a lab? What did you say there? Because that's a minefield of its own, right?
That was quite a while ago, actually. I might have to double check that. But yeah, essentially it was, it's difficult to make a virus without having something to start with. We have these already well-established like viral strains that we can edit within the lab, and the coronavirus doesn't match up to anything that's been previously published on any of these like modified viruses. So essentially what it comes down to is it's highly unlikely that it was like engineered and intentionally released in a lab. Like obviously lots of people within the Wuhan region are working on coronavirus because it's prevalent there. So they have the bat populations not far away. So it's still possible that it's maybe got out from a lab. But I would say it's almost certainly not like a supervillain releasing a virus upon the world. I think part of the article, I actually have a supervillain interlude, and it's like, if you were a supervillain, why would you bother doing it with this virus? Because it's just, not the. Well, you wouldn't think it would be the world ending, but I guess it has ground the world to a halt in certain times.
That's right. You need if you're ever going to say that again, we need like the supervillain music. Yeah, I read that too. Most scientists that I've talked to, they can't rule out that it didn't come out of a lab. But like you concluded, it's very unlikely that it was made in a lab. Okay, I was just super curious for your take on that. That's all. So the two more questions I have is you're gonna have to talk everybody through this. You make movies of the proteins in action.
Yes.
How is that even possible? Like, this is very small. Like,
yeah. So with like the single crystal we described, and you've got the picture up of, that's kind of a static snapshot of a protein in a crystal. So kind of one of the big downsides of protein crystallography is a lot of it is done at cryogenic temperatures, and that's to save the crystals from the massive amounts of radiation that we expose them to.
X-rays.
Because that obviously, yeah. Yeah, so that damages the proteins, and then like the diffraction gradually disappears as the radiation breaks down the crystal effectively. Mm-hmm. So to make movies, for one, we need to do this at room temperature, which means the crystals take a lot more damage quicker. So we actually use a different technique. It's called serial crystallography. So instead of collecting one complete sweep from a crystal by rotating it in the beam, we collect like thousands of single patterns from crystals in random orientations, and then combine that to get our complete data set. So that's how we get a room temperature structure without doing too much damage to it.
And then you put it together like, oh, I'm sorry. Go ahead. Sorry. I've got a bit of a lag. I apologize. I don't mean to talk over you. I'm so sorry.
Sure. No problem. Yeah. So that's how we'd get like our what we call our dark state, like the resting state of the protein. And then what we'll typically do is we'll have some means of activating our reaction. So we'll either be mixing the thing that the protein wants to react with it by firing like a small droplet of it, or a lot of the time we have the ligand that the protein wants to interact with caged in what we call a photo cage, and we'll fire a laser at that to release the molecule from the cage, and then it can start reacting. And then what we'll do is we'll collect these structures at different time delays, and we then combine all those different time delays to see the reaction happening across the different timescales.
It's unbelievable. And
it's pretty cool.
And then you just put it together like a claymation thing.
Yeah, pretty much. Yeah,
like
so we
Wallace and Gromit.
Yeah, so we have a piece of software.
I'm sorry.
Yeah, so there's a piece of software called Pymol, which is just like a viewer for your protein structure. And yeah, we can just make our figures from that, and you can get it to like morph from one to the other, and you can make your molecular movie.
That is amazing. In the nest, I've put the link. Excuse me. In the nest, I've put the link to InsideCorona.net, and I'm mesmerized by the little looping video of the coronavirus on the landing page.
Yeah, that's
terrifying. It's so scary.
Well, that was actually part of the reasons we picked those color schemes, because we didn't want the coronavirus to look nice.
No.
Like, it's meant to be the villain in this story.
Well, it is. It sucks. Stupid coronavirus.
Yes.
Okay.
Yeah, I've managed to avoid it so far, but my partner has just caught it, so I'm probably not far behind.
Oh man, our family, all like my wife, myself, and my son who still lives at home, we haven't got it, but we've all I've gotten sick, but I've tested negative, and so has my son. I don't know. We'll see.
Yeah, I'm still testing negative so far, so maybe I can fight it off.
Well, good luck with that.
Thanks.
That's fascinating. Doc, is there movies? Like, are those things that you make available to the public anywhere? Like, I'm just curious what they would actually look like when you put it all together. Do you know what I mean?
Yes, I mean, you can. So obviously we publish in journals is probably the standard way we like disseminate our movies. So yeah, I can probably find one that I have published somewhere.
Well, if you find it and DM it to me, I can post it and put it up in the nest for people to take a look at. I have to get off that landing page on InsideCorona.net because that I do not want to stare at that coronavirus anymore. I hate it. Our province of Alberta made this mascot called Uncle COVID or something. Chris, what was his name? You know that terrifying coronavirus guy?
It was Uncle Mike.
Uncle Mike? Yeah. Anyways, it was a guy, and they had this like silicone coronavirus face that was terrifying, and they made the eyes blink, and it had sharp teeth, and it's like coronavirus Mike is coming, or Uncle Mike is coming over, and he like shows up, hello, and he gives everybody coronavirus, and it was supposed to be a bit tongue in cheek, but everybody was terrified of coronavirus. Uncle Mike. What?
Yeah,
was
Uncle
it? Uncle
Mike
Mike
is coming for Christmas. Uncle Mike is coming for Thanksgiving.
Are you sure it was Uncle Mike?
Yeah, but I could check. I'll check.
Yeah, what a terrible uncle.
Well, I'll see if I can. It's creepy Uncle COVID. It's Uncle COVID, Chris. Oh my goodness.
I thought his name was Mike.
Well, COVID loves the holidays. Okay, I'm going to post this and then throw that up in the nest for everybody to take a look at. This is what the Alberta government used to try and stop people from gathering in massive groups. To stop, I believe it was it the Omicron variant? Something was going. Anyways, were you able to find the paper, or should we just continue, Doc?
Yes, I DM'd it to your.
Oh, sweet. Okay.
Bunsen and Beaker.
Perfect. So I'll ask the next question, and I'll set up that stuff in the nest for people to take a look at. That way I can multitask. So if there's time, we usually ask our guests to share a pet story. Now you were like, I don't know if I got a pet story, but you actually replied to the advertisement for this space about a ferret. And yes. I have to tell you, this will be the first ferret story in the history of me doing a podcast or all of these things live. And I am so excited for your ferret story. Take it away, Doc.
Yeah, so there was a slightly different synchrotron to the one I work at. It's actually a proton synchrotron. So it is actually one where they collide stuff to see the building blocks of atoms. But when they were building it in, I think it was the 1970s, this guy had said to the government, like, if you give me $250 million, I can get it working within 5 years.
Okay.
And they were at year 4 and it wasn't working.
Uh-oh.
So they were trying to get the protons going round the ring with all the magnets, and they were trying to get it up to the energy they wanted. But the magnets kept shorting out.
Uh-oh.
So eventually they figured out the reason that the magnets were shorting out were there were these little slivers of metal still in the vacuum tubes where the particles were trying to go round.
Right.
So they had to figure out a way to clean the inside of these tubes and get these slivers of metal out. And it was actually an Englishman from Yorkshire who was working in the States who suggested using a ferret because ferrets were quite well used within the UK for like hunting rabbits and stuff, because they'll happily go down a hole and flush out rabbits and stuff like that.
Okay,
so the idea...
that's what they're used for in the UK? Like people go use them for hunting rabbits?
I mean, you can do. You can also just have them as pets. I remember actually... That's
bananas.
I remember as a kid, actually, we went to like a local fair, and they actually had ferret racing.
What?
So you pick a ferret, and the breeders or whatever, they hold the ferret at one end of the tube, and you hold a bit of raw chicken at the other, and then they release them, and then it's whoever's ferret reaches the other end first wins. This is what it's like growing up in the countryside in the UK. It's just a bit weird at times.
Oh, we live in rural Alberta. Talk to me about weird stuff that happens at fairs and things like that.
Yeah, I mean, I grew up in not even a village, a hamlet of 400 people.
Oh my goodness!
So really, really tiny. But yeah, back to Particle Accelerators. So this Yorkshireman came up with the idea that they could use a ferret to clean the inside of these vacuum tubes. So they bought a ferret from a local, I think it was a mink farm, and she was the smallest ferret they could find. I think she was 15 inches long or something like that. And they named her Felicia. And when they tried to get her to go into the 4-mile-long tunnel, she wouldn't go into that one, but they did eventually manage to train her to go through the smaller tunnels, like the shorter sections that hadn't been linked up. Actually, one of the other funny things is so what they wanted her to do was they'd have like a collar on her and a bit of string, and she would pull the string all the way through the vacuum tube to the other end. And then once she'd done that, they'd attach a swab with like a cleaning solution on it and pull the swab through.
That's what you use with a sewer snake. But
the other thing is, obviously, ferret feces or urine is also not very good for X-ray vacuum tubes, so they also had to put a little diaper on her.
Oh, well, that's cute.
Yeah.
Now, I'll have to. Okay, so I'll find. It will be on your profile, the picture of this ferret, right? Because you tweeted in response to the ad about this. So I'll throw it up in the nest for people.
Yeah, if you want the full story as well, I can send you a link to the Atlas Obscura article.
Okay, I'll have to keep looking. I can't find it. Okay, it's okay. It's an adorable story. We can. While Dr. Sam was talking, I've put up in the nest a YouTube video of Uncle COVID from Alberta. For everybody, I'm already getting DMs. This is nightmare fuel. Why would you post this? Why did they think this was a good idea? And then also the journal that you published in, which I believe if you click on the link, it takes you to the journal, and then somewhere at the top it says video. I think if you click on that, the video comes up. Is that Am I on the right spot there? Because it is pretty. There's a lot
yes.
of stuff.
Yeah, sorry, I thought I'd sent you directly to the video page, but possibly not. Yeah, if you click the video, then it should.
There's like an icon. There's an icon for video, and then yeah. I'm super curious.
Yeah,
I'm gonna watch this right now. I'm sorry, just a second. Oh my goodness, look at it go.
Yeah.
Okay. Oh, look at them. They're jiggly. Okay.
To NO.
Sorry, did my audio cut out? I was watching your video. Sorry, I didn't mean to talk over you. Go ahead. Sorry.
Sorry. So this is a protein called nitrate reductase, and it's a copper protein. So it produces these beautiful green crystals because of the metal centers. And what it does is it converts NO2 to NO by transferring an electron between the two copper sites. And then there's also, if you see just above the NO2, there's a aspartate amino acid, and that then transfers a proton and creates water and then leaves NO. So we made this movie, and it kind of answered a question that had been argued about between spectroscopists and structural biologists for a very long time about the orientation of the NO2 relative to the copper. So yeah, kind of like a basic research thing. Like obviously no pharma company is super interested in how NO2 becomes NO, but like for us it was super interesting.
I love it when scientists have beef with each other over something the general public doesn't even know they're having beef over each other with. That's my favorite. That's my favorite thing.
Yeah, tens of years of conflict of like spectroscopists saying like, but our data says it's end on, and then like, but our structures say it's face on, and it's like, but this says it's end on, and then they'll publish another paper, and it's like, but look here, it's face on. And it's just like back and forth and back and forth.
And you're like, no, we've got the receipts. Look at the video. Check this out, nerds.
Yeah, exactly. Yeah, so we also did this at a couple of different temperatures. So we did one at cryo temperatures, one at 190 Kelvin, which is like this glass transition temperature, and then one at room temperature. So we can see sort of all the dynamics at the different temperatures of how this reaction works.
Nice. Thanks for sending me the article about the ferret. That's up in the nest for everybody to take a look at.
Nice. Yeah.
Well, I think it's time to open up the floor to questions from our community.
Okay.
I don't know how active you are on Twitter, Doc. There was a massive fight 2 years ago between the bird scientists and the fish scientists. Like they had serious beef with each other for about a month. It was hilarious.
I've played D&D with both a fish scientist and a bird scientist. You know, Dagmar Duwedderworth.
Yeah, I know Dagmar.
Alex,
Dagmar.
Alex
There we
Evans.
doing. Yeah, Dagmar. And who's the other one? Sorry.
Alex Evans.
Alex,
he
you know
runs
Alex? What?
Yeah. We've played D&D together live on stream for charity.
Those are amazing people. I can't believe you know those two people. That's so cool.
Yeah. Yeah, they're good guys. Yeah, very fun to play. They're absolute chaos in D&D, but yeah, great fun.
I can imagine. They both got very strong personalities.
Yeah, Alex's character was a definitely not copyright Mothman. Definitely not Batman. Definitely Mothman.
Mothman.
Yeah, it was super fun.
Was Dagmar's an archer?
No. Dagmar
Okay.
was a Lothario bard type with a very fine pencil mustache.
And what's your character, if you don't mind me asking, Doc?
Oh, I was the dungeon master.
So you didn't have a character. Oh, okay.
Gotcha. I was the villains.
You were all the villains. There you go. Well, that makes sense because you work on the coronavirus. You can
exactly. Yeah.
It all comes full circle. All right. So for folks that are asking to come up to the stage, Again, we do check your profile to make sure you're following us, and we check to see what you've been tweeting. We do try and keep this space safe. I see Indra and Liz are up here, and we'll keep our questions probably on point with what you're talking about with proteins and maybe the coronavirus even, and even D&D.
Yeah, I should preface this with I am not a medical doctor, so I will not be giving any medical advice.
Right. Okay, so we'll go to Indra, then Liz, then Danielle. Hi, Indra.
Hi, Jason and Chris and Dr. Sam. Thank you so much for hosting this space.
Happy to be here.
So I found this really interesting, and I was making dinner. I'm always multi-skilling when this space is on. And Dr. Sam, I did actually, like, my ears kind of pricked up when, I was picking up all sorts of different things, but I was interested when you mentioned that there was, with the protein studies and the movies. And I make different types of movies. So I was just so fascinated to know about the details. So I was trying to absorb it all. But from your perspective and studying the proteins, does that make it, and I know you're not like a medical doctor, but it sort of sounded, and I guess I'm also coming from the fact of, from a nutritional point where my mum's a nutritionist, and
Sure.
she always kind of enforces the importance of eating enough protein in your diet, particularly if you're very active. So I was trying to just see the correlation between the importance of protein in the human body and, with the science of what you were saying, that it could help, with people and their health overall. I was just curious, do you feel like it is important for us to sustain a certain amount of protein in our diet?
Yeah, I'm obviously not a nutritionist, so you're probably more of an expert on how much protein we should eat. But in terms of the body in general, proteins are the workhorse of our body. If there is a process going on in our body, it's probably done by a protein. All of our respiration, all of our ways of creating energy, breaking down our food, a lot of it depends on proteins. So yeah, I guess eating enough protein so we can make more proteins is probably the important thing to think of.
And sorry, just a quick follow-up on that. And so if your research into the nature of proteins and how they operate with our human body, so would you say that, that can help, with all sorts of different diseases maybe in the future? Is that what you're looking into? Because I only picked up a little bit of that.
Yes, most using cancer probably as an example, most times when someone ends up with cancer, a lot of it is a protein has gone wrong, and that disrupts something within our cell processes, which makes the cells divide in excess. So a protein within a long chain of proteins goes wrong, that messes up the homeostasis of our cell processes, and then suddenly we've got one cell that just won't stop dividing, and then that makes a tumor. So a lot of the big problems with treating cancer is 5 people can have the same cancer. You can have a bowel cancer, but each one of those 5 people, that cancer might be caused by a different protein in a long chain of proteins. So to be able to specifically treat a certain cancer, we need to know which protein is going wrong. And then once we know which protein is going wrong, then that's when we come in, where we can solve the structure of that protein, and then you can design a drug that fits kind of directly into that protein to stop it going wrong.
Wow, that sounds amazing. Well, thank you so much, Dr. Sam. And I'm going to head up to the nest to watch some of your movies, because they're certainly different to the movies I make. And thank you so much, Jason and Chris.
No problem. I wouldn't watch that Uncle COVID one before you go to bed. You'll have nightmares.
Yes, I was going to abstain away from the COVID one. Yes, thank you for the pointer.
Yeah, I might watch that one after I wake up later today.
Once again, I am Han.
And I am France, and we are here to pump
You are.
There we go. There's some good protein. There's a protein sound effect for everybody. Yeah, it's fascinating, right? And that is so true on point when you look at all of the different cancers. Just the smallest error can cause the biggest problem. And I never thought of it that way, that your technique could potentially weed out what the problem is right down to the curlicue that went wrong. So,
Yeah, so it'll be like the molecular biologists will probably figure out like, oh, this protein is going wrong. And then once we know the sequence of it, we can express that protein like in some E. coli cells, purify it, crystallize it, and get the structure of it, hopefully.
Nice. And then from that blueprint, everything else is built. Very cool.
Exactly.
Very cool. Yeah, you're like the blueprint that builds the house. I'm getting it now.
Yeah.
Over to Liz, then Danielle.
Well, good evening, everybody. Hey, Doc, I have 2 questions. They are from a friend. They literally are. They're from Rachel, and she would like to know kind of what your thoughts on monkeypox is. Do you see it as being potentially as prevalent as COVID? And then a follow-up, what do you see as the biggest challenge in your field these days?
Yeah, so I'm not massively well read on monkeypox. The only reason I actually know about it is there was a terrible conspiracy theorist who I came across on Twitter who was tweeting a load of stuff about monkeypox, and I just had to go and read up on some stuff and find out if what he was saying was in any way correct.
They're the worst.
And yeah, he was way off. But yeah, from what I've read, the current spread of monkeypox seems to be very focused within actually sexual circles. It seems to be spreading between mostly men that have sex with men. So it's nowhere near as like infectious as COVID, which is just an airborne particle. So I don't think it's likely to spread like the same way that COVID has. It seems that its method of spreading is nowhere near as virulent. It's also quite treatable already with the vaccine we already have. Was it? Which vaccine was it?
I did read that. I forget the exact vaccine.
Was it the polio vaccine?
I want to say.
No, the smallpox vaccine.
I was going to say I was going to say a smallpox vaccine.
Yeah. So I think it's definitely not going to reach the kind of levels that COVID has because we already have a viable treatment to prevent it spreading. So yeah, that shouldn't be too much of a problem. But again, I am not an epidemiologist or an expert in this. I can just tell you what the proteins look like in the monkeypox once they've been produced. And what was your second question? Sorry, I've completely forgotten it.
I forget.
Oh, no. She was wondering what the biggest challenge in your field you see nowadays.
So yeah, I think the big thing for us is actually there's new techniques that have come along. So cryo-electron microscopy is actually being used a lot to find the structure of proteins now. So crystallography is kind of having to go through the extra effort of crystallizing our protein. A lot of proteins also can't be crystallized, so it's really difficult for us to work on like these really, really big proteins. So that's where cryo-EM comes in. So crystallography is kind of having to find its niche alongside cryo-EM to kind of stay relevant, really. And I think drug design and the sort of dynamics work that I'm working on, like really getting into like the nitty-gritty of how these individual proteins work, is probably where the field is going at this point. Yeah, I'd say that's the main challenge. Yeah, finding our new niche in a changing life of new techniques.
Well, awesome, Sheena. Thank you.
Great questions, Liz. And by Liz, I mean Rachel. So it was How did Rachel get her question to you? Did she email it, Liz?
Oh, we just been DM.
Oh,
right, so.
okay. All right. I did go down the hole of monkeypox like 2 weeks ago, and like a bunch of epidemiologists on Twitter were talking about it, and from what they said is you have to be up and close and personal with somebody for a bit of time to get to have it transfer instead of like coughing on somebody. Yeah, that is what they said. It's not near as contagious. So yeah. Okay, over to Danielle. Hi, Danielle.
Of course, there are already people saying that
Oh, sorry. Go ahead, Doc.
Hey, how's it going?
Oh, hi, Danielle.
There's already people saying that monkeypox isn't real.
Oh, well, neither are birds or Australia, apparently. Okay, go ahead, Danielle.
I mean, frankly, for all the animals that seem to be trying to find creative ways to kill humans in Australia, I would be okay if they didn't.
Oh, you cut out a bit there, Danielle. Could you say that again?
So, So, Doc, first off, thank you for being. Oh, I'm sorry. Is it you can't hear me? Hold on a second. Oof, I've been having issues with my audio all night. Actually, can you hear me?
It's really good now. Yep.
Okay. Well, first off, Doc, thank you for this. I have learned so much about a topic I really didn't even know existed to the depth that you were talking about tonight. So that was incredible. But I know, honestly, that image that I cannot get out of my mind is a ferret running in these tubes with a diaper on. And I just want to know, is she the only ferret working in her field, or are other labs using that technique?
I think since they have found a better way of cleaning the tube. So we currently don't have any ferrets in employ at Diamond Light Source. And unfortunately, Felicia was working in the 1970s, so she is no longer with us. There was actually a section in the article I was reading, and they planned to get her stuffed and put on display, but she went missing, so they actually have no idea where she ended up.
Oh my goodness.
That story was a roller coaster, and I thank you for that.
I know, it's got everything, hasn't it?
Oh my goodness. This is outrageous.
Thank you. It wasn't necessarily a serious question, but that was just, when I heard that fairy story, that just lodged in my mind. I had to know more.
Yeah, somebody get that ferret a Nobel Prize.
Standing ovation for the ferret. All right. So we have a question from DM. It's from Cobalt. Mm-hmm. Cobalt was asking about if you've taken pictures of peanut proteins to figure out maybe how to make them not attack somebody's system. There's a lot of people that are really, really affected by peanut protein allergies. Is that anything that you've come across?
I have not personally, but I've just gone to the PDB and typed in peanut, and there are some structures of peanut protein allergens. So there probably are people working on it from the looks of things. But in terms of modifying it to make it so you're no longer allergic, I think that would be very difficult, because you'd have to edit the entire genome of the plant that produces the peanut itself. Well, which, I mean, we can edit genes in plants, so it's probably not beyond, but I think so that the way the immune system works is it's like if a foreign protein gets into our cells, that protein will be chewed up, and then it's displayed on the outside of our cells to show that it's like a red flag to say that like something is wrong here. So I think it would be a Herculean task to be able to edit the genome to the point where the protein no longer produced an immune response, which is very sad for people that can't eat peanuts because peanuts are delicious. Like, I am glad I'm not allergic to peanuts because I love anything satay, anything with peanuts in. It's great. I'm sorry I can't help.
Well, I don't know. I think you're selling science a bit short. You did just finish explaining that you trained a ferret to go through a pipe to clean out stuff. So I'm not sure.
Yeah.
I'm not sure anything is out of the realm of science after listening to the ferret story. So, yeah. Well, you never know. With CRISPR in the future, there might be a way to genetically modify that entire strain of peanuts to get rid of that protein. Or maybe that is
it's possible.
It could be a pipe dream. You're right. Yeah.
Yeah, I think I don't know too much about the molecular biology between like how the peanuts cause the allergic reaction. If it's a single protein and it's a protein that can be knocked out, then yeah, maybe. But I think it's probably going to be like a lot of different proteins within the peanuts, all that could cause an allergic reaction. So it's whether if knocking out all of those proteins like just stops the plant being able to function.
All right. Gotcha. Hopefully that answered your question, Cobalt. Thanks for sending that by DM. It's kind of tricky sometimes to answer questions through DM. So we're coming up to the top of the hour. It is really early in the morning for Dr. Sam. So I think just to give you a break, we'll wrap up the space now. Make sure you follow the good doc who's up top there. If you click on Dr. Sam, you can come up to see his profile. It looks like you've got some kind of like banner holding some green crystal staff. What's that about? Can you talk to us about that before we shut her down?
Sure. So it's from it's a website called Hero Forge where you can like make a miniature of yourself.
Oh, cool.
Or a miniature of anything. So I just made myself a crystal wizard and then put some diffraction behind it.
Oh, because you're a crystal wizard in real life. I get it.
Mm-hmm.
I get it.
Yeah, and if you scroll down a little bit on my profile, you'll also see a pinned tweet of all of the protein crystals pictures that I've taken over the years.
Mm-hmm.
And some of them are absolutely beautiful. I would really recommend having a look at those.
I use one of those photos in the ad for the show. Yeah, they're just so cool. Science is amazing. Okay. Just from me to you, thank you so much for being our guest tonight and on short notice. I think I contacted you like a little over than a week and a half ago and you said yes. And you said yes to doing this at like freaking 2:00 in the morning for you. So, everybody
thank you for having me.
Yeah. Everybody give Dr. Sam some love in the space, with some emojis of hearts or claps. If you're on Wisdom, you can give some emojis too. Oh, people are giving you emojis on Wisdom. Come on, Twitter Spaces. You got to do better than Wisdom. And we had 2 people in Clubhouse. They're gone now. So it's just me talking to nobody on Clubhouse right now. All right. So the hearts are coming in. When you wake up in the morning, are you heading into work? Do you got tomorrow off? What's going on? Are you going to be able to have a late day?
Yeah, we work flexi time, so we're actually one of probably the few science jobs where I actually do end up being on call overnight. So like,
oh, what?
We didn't really get into this too much, but as a beamline scientist, so I help run one of the beamlines where users from all over the world come and use the facility to collect their protein crystal data.
Yeah.
So obviously that is a very finely tuned machine that can go wrong at any time. So we're actually on call like once or twice a week, and it's 24-hour operation. So if something goes wrong at 3 a.m. and we're on call, we get a phone call and have to wake up and log in and try and fix the problem. So
what?
This is just like being on call. So it's not too bad really.
I would get up at 3:00 in the morning for a dog. But if one of my students was to email me at 3:00 in the morning, I would delete it out of spite, I think. So you're a better person than me.
Yeah. I mean, we do get a little extra pay for receiving a call, but if something goes wrong early in the morning, you wake up bleary-eyed and try and fix part of the particle accelerator for people.
Oh my goodness. Well, again, thank you for being our guest tonight. I've learned so much. I did try to do some background, and I just bamboozled myself. So I'm so glad you were able to explain what the heck you do and how important it is. I think the takeaway is all of the stuff that you do builds the blueprint for future protein research. Because without knowing what it looks like, you can't do much.
Yeah, I think we definitely feed a lot of information into future studies, and I think that's really, really important. A lot of basic research doesn't kind of get the accolades I think it deserves. Like a lot of the really, really high impact Nature papers are built on a lot of basic research that doesn't necessarily get the big press release.
This is so true. Yep, that's so true. All right. Well, I think we'll wrap things up. I'll let you get to sleep. Thank you again for waking up blurry-eyed to help us in Twitter Spaces instead of a particle accelerator. So we'll just wrap it up. Thank you, everybody who
Thanks
came.
for having me.
Yeah, thanks so much. Make sure you give the doc a follow, everybody, and check out all the stuff in the nest. I've recorded this space. It's going to stick around for about 24 hours, and then it goes away. Our patrons, the Paw Pack, get a high quality recording right away. Dr. Sam, I'll send you a link to that recording as well. And and if you want more spaces that we run, every Saturday we run Pet Chat at 6 p.m. Mountain, 8 Eastern. It is definitely lighthearted and very wholesome. We play games and talk about dogs and cats. July is going to be a very busy month. There's a whole bunch of extra spaces that we're going to be running with dog trainers and extra scientists. I'm really excited. Early in July, we're talking to Emily Calandrelli, also known as the Space Gal. Emily had a Netflix show called Emily's Wonder Lab. She's on Fox News, The Inspiration Outer Space Station or something like that. She's got almost 1 million TikTok followers, and she's an awesome mom scientist. So we'll release information about that. Chris, thanks for being the silent person behind the scenes, getting everybody up to the stage. I don't know if you can.
No problem.
Yay.
I can. Yay.
And for everybody listening on Wisdom, we had almost 70 people on Wisdom, a couple people on Clubhouse, and the 40 to 50 odd people that tuned in here. You could be anywhere in the world and you're listening to us talk about science with amazing guests. Thank you so much. And once again, Doc, thanks for being here. Take care.
Good night.
All right. For science, empathy, and cuteness, everybody. Space closing in 3, 2, 1. Take care.